Oh, welcome, everybody. I was just looking, and we're still having a few people join. Merit Cudkowicz, happy Thursday. I have the honor today to present our guest speaker, Dr. Robert Bowser, known to many of you. He's the Chief Scientific Officer, a Professor and Chair, the Department of Translational Neuroscience at Barrow Neurological Institute in Phoenix. We refer to him as the biomarker guy, but he really is leading all the many initiatives and is the go-to person for the discovery of biomarkers in ALS. He's going to talk to us about his work and the All ALS Clinical Research Consortium. Welcome, Bob.
Thanks a lot, Mary, and thanks for everyone for joining today. And yeah, it's a very short presentation. I'm going to take you through a few slides, a couple of examples of biomarkers, and really some generalities around biomarkers, and then some initiatives that are really critical for the advancement of biomarkers. So next slide, please. So what are biomarkers? I think most of the people on board have heard of that. And as Merritt said, I've been doing this for a while before it was sort of in vogue, and leading a bunch of efforts around biomarkers in ALS. So where do we look for biomarkers? Sources of biomarkers? Great ones, obviously genetics, genetic mutations, risk factors, genes that are causative of the disease. And so these are fantastic sources of biomarkers to identify individuals that have a particular type of ALS or may be predisposed to form ALS. A lot of biomarkers come from biofluids, so cerebral spinal fluid or blood, urine or saliva, looking at proteins or metabolites, other aspects that we can identify as biomarkers that are related to maybe the onset of the disease, progression of the disease, et cetera. Tissue, taking muscle samples, skin samples, and incredibly valuable is postmortem tissues. Really looking at tissues from patients with the disease and then asking questions around some of the biomarkers that we find in biofluids, what cells are expressing them within the nervous system and around the areas of disease for ALS. Digital-based biomarkers, speech, movement, other digital devices that we can use to monitor patients and evaluate aspects such as speech, and imaging. So imaging biomarkers can be important as well with both PET, MRI, DTI, different flavors of imaging. Next slide. So what do we do with these biomarkers? What are the goals? And so if we start up on the top right, so advance it forward. So the right target. So next. Yeah, so the right target, a particular gene mutation, or maybe some receptor on a cell surface that we've identified as a biomarker, maybe a downstream biochemical pathways that might be involved in the disease. So what are the right targets to hit? The right drug, next. So using cell-based models, it could be induced pluripotent stem cell-based models, animal models, to demonstrate that a drug of interest actually hits a target. And that's developing biomarkers that are called pharmacodynamic biomarkers for whether or not a drug of interest is hitting its target that then might be used in clinical use. The right biomarker, next. So for clinical trials, obviously genetics or pathways for inclusion can be really critical for the right type of biomarker. I just mentioned a pharmacodynamic biomarker demonstrating the drug's hitting its target. Or a biomarker that responds to a drug treatment that tells us whether or not the drug is having an impact on a patient. Next, the right participants, obviously as early as possible. And now we can even think pre-symptomatic in those that are carrying specific gene variants that we know are potentially causative or risk factors for developing ALS. Or maybe stratifying the patient population, either maybe fast progressors versus slow progressors, or enriching for a patient population that has, say, an activation of a pathway that a drug of interest is targeting. And then the right trial. So can we use biomarkers to reduce the number of participants required to really power statistically a clinical trial? Can we use biomarkers during a trial to maybe reduce the time for making go, no-go decisions? Or maybe using biomarkers as new outcome measures in clinical trials. Next slide. And again, the key is really developing a biomarker toolkit that can really inform us and help make decisions in all the steps of the drug development process. Next slide. Not going to go through all this, but really just showing you, here's our cells of interest within the central nervous system, motor neurons, astrocytes, microglia, the muscle with neuromuscular junctions. All of these listed here are biomarkers of interest that have been identified by my lab, lots of labs around the world, as relevant in ALS and of interest in looking and trying to either identify the disease, monitor progression, or look at treatment responses. I'm only going to focus on one of these because it's a short presentation and there's lots of them. The one I'll mention it further, a little bit further, is neurofilament. So next slide. So neurofilament right now is a top protein-based biomarker for ALS and also for frontotemporal dementia. So that comes into play related around C9-related diseases, which can go either towards ALS or FTD. So neurofilament's expressed in neurons in the central nervous system, highly abundant. It's a protein that is involved in maintaining the architecture and the framework of that neuron. And then under times of injury or during damage, the neurofilament protein can be released from the cell. And that can accumulate initially in cerebrospinal fluid. And we can use assays to measure it. And then ultimately it goes out into the bloodstream and then ultimately gets eliminated from the body. But then we can use assays also to measure it in the blood and then correlate those levels to a disease state or treatment, for instance. Next slide. And so thinking about a protein like neurofilament, in this case, neurofilament light chain, is a prognostic biomarker. So a number of investigators around the world have looked and sort of correlated levels of neurofilament, both in the cerebrospinal fluid, which is shown on the left, and the levels of that correlated with the rate of disease progression. So if you had the highest levels of neurofilament, that's in the red, the rate of disease progression was faster. And if you had lower neurofilament, your rate of disease progression was slower. So it's providing some input in correlation between rates of disease progression. And then a number of studies have been looking how early during the course of disease do you get an elevation or a rise in neurofilament. So neurofilament levels are not specific for ALS. They're increased levels in other neurodegenerative diseases and other injury states of the central nervous system, but they're actually pretty high in ALS, even compared to other neurodegenerative diseases like Alzheimer's disease. So how early do they occur? And so there's been a couple of studies looking at this and looking at patients that were either symptomatic or asymptomatic. This from a German group showing that, well, we really didn't see it too much in the asymptomatic patients. But then the next slide, please. In a further study, now looking longitudinally, so that what I just showed you was really looking at what's called a cross-section, one time point. So an asymptomatic person at one time point. Here, what I'm showing you is a small group of people followed longitudinally over time. And so the x-axis is time, y-axis is the amount of neurofilament protein. And what was identified in this paper was that even before symptom onset, which is what's listed in the x-axis as 0, so time from onset of clinical symptoms, so that's the 0 time point. So you can see in a number of patients, neurofilament levels were above what is considered sort of the average normative value, and that's the whole gray zone there. So if you're above the gray zone, you're above normal. And so in patients that were even asymptomatic clinically, they already had elevated levels of neurofilament, which upon becoming symptomatic actually increase even further. So potentially this is occurring very early in the disease process and therefore a very important biomarker for ALS. Next slide. So can we use this in drug trials? And it's currently used in a number of trials. And I think almost every trial ongoing is looking at neurofilament as a biomarker. On the left panel here comes from a study from Biogen where they were treating patients that harbor genetic mutations in the SOD1 gene with their antisense oligo, so ASO, to decrease mutant SOD1 expression. And in the red were patients that were provided drug initially, and you can see a decrease in the level of neurofilament, which remained constant over time. And there's a dashed line, vertical line, And that was the length of the clinical trial, 28 weeks. And then they did what's called an open label extension. So they provided patients, even that were on the placebo group, which is the blue, with the drug. And what one saw was that once even the placebo patients received the drug, they also showed this great decrease in neurofilament over time. And this was really used by the FDA to help get accelerated approval for this drug. Now, if you look to the right, this is data from another trial from Biogen, looking at an antisense oligo treatment to C9 types of ALS, so that repeat expansion. And what they saw here was that when the drug was provided, they actually saw an increase in the levels of neurofilament over time when compared to the placebo group, and that increase was dose dependent. Clinical parameters of the disease, so outcome measures clinically, were also not improving, and in some cases getting worse. And so based on this information, Biogen looked at this increase in neurofilament and terminated the study and said that this is probably ineffective for ALS patients. So this is the use of neurofilament as a response to drug treatment and the value of it in clinical trials for ALS. Next slide. Now, there's lots of important questions that remain. So I showed you in one case where drops in neurofilament help lead to FDA approval, but exactly what is that percent change that is required in order to demonstrate you're really impacting that pathway, that target pathway of interest? And what percent change actually correlates with positive clinical outcome measures? And I think we're going to continue to learn more and more about how neurofilament levels, as we measure them in blood and CSF, are correlative with clinical outcome measures, and then what percent drop in neurofilament really helps to demonstrate that, yes, your drug is hitting target and doing what you hope it's doing in patients. Next slide. This is just another example. I published this out of my lab into last year. And here we're taking, in this case, again, longitudinal CSF. In this case, we use samples from just fast progressors or slow progressors. And the question was, what's different? What is different biologically in people that are progressing rapidly in the disease versus those that are progressing slowly in the disease? We used a technology called mass spectrometry-based proteomics. And the squiggly lines you see there, you can see fast progressors, and this is months since the first draw, so 0 to 30 months in a longitudinal measure. Each different color is an individual. What you see is, in an easy way to describe it, sort of a noisiness, an entropy of the system, and of all the proteins that are present in the CSF. If you look at the slow progressors, you see less noisiness, less entropy. So it looks calmer within the patients. And we think, my hypothesis is that if you're a fast progressor, there's a lot of things ongoing. You have other immune cells coming in from the periphery, a number of pathways that are activated and changing over time in fast progressors. But in slow progressors, you're seeing that at a much lower scale. And so you have a much calmer, in essence, proteome. Next slide. And then with some physicists in California, we took the data and created a mathematical model. And so we could use just one time point, one CSF sample, and then use the model and predict, are you a fast or slow progressors? And then we can move that over time. And so what this data is showing is that, again, this is the same data that I just showed you between sort of 0 to 30 months, but now using a mathematical model, we projected backwards in time and said, when do we start to see these changes in sort of the variability of the proteome? And what the model predicts is that in fast progressors, you see this very early in the course of disease. And so it could be a way to monitor and track people quickly, whether or not you're a fast or slow progressor. And then in a clinical trial, could we take this sort of fast progressive, high level of proteome entropy that you see on the left and change it into a slow progressing form? And so I just showed you data on looking at a protein, neurofilament, and its value in clinical trials. In this case, we could be asking, could we look at all the proteins? Why look at just one? Let's look at everything and say, can we show or demonstrate an impact of a drug looking across all the proteins in the CSF? Next slide. So again, just harping on something that's really critical is that, we have an urgent need for continued research on ALS biomarkers. I've showed you some of, the best biomarker to date, data around another methodology, looking at all the proteins. But the goal is, we need this toolkit. We need a variety of biomarkers, and we need, there's an urgent need to continue this type of research. What that requires is continued participation of individuals in clinical research studies to collect biosamples for research purposes. And I've just listed a few currently enrolling ALS clinical research studies that are collecting samples in sort of a natural history types of studies. And, Target ALS, ALS TDI, the CDC registry and biorepository, everything ALS. And then a few of these asymptomatic gene carrier studies that are following patients that harbor gene variants that are causative of the disease. So these are really critical studies. And now we have another upcoming one, next slide, which is called All ALS, which is Access for All in ALS. Next slide. So this was an initiative that is now received funding from NINDS through the Act for ALS funds. And myself at the Barrow Neurological Institute and James Berry at Mass General are sort of the contact PIs for a very large group of individuals in a number of academic centers that are generating a national ALS research consortium. So again, this was just funded in October. Our goals are creating a very large flexible research consortium platform that can grow and be modified on where the research is moving. The goal is to provide opportunities for everyone living with ALS in the US to participate, running this natural history and biomarker study, collecting samples and openly sharing them, providing clinical data and samples again to researchers around the world, and hopefully identify new, better, improved biomarkers to aid drug development. And our goals will keep expanding as we move forward. Next slide. This is sort of an overview of this consortium, and above it, you see this Act for ALS public-private partnership, and there's a steering committee called AMP ALS. The AMP ALS steering committee helps guide the consortium underneath that, and again, we have two coordinating centers, one on the west at the Barrow that I'm leading, and the one on the east at the HEALEY Center at MGH that James Berry is leading. Sharing protocols, sharing everything, one single IRB. We collectively have 34 sites that are starting, and the goal is collecting samples in a biorepository and having a data portal to share all the information. Next slide. Listed here are just the 34 sites. Again, not going into individual sites, but trying to geographically distribute them around the country. Next slide. But we have fairly aggressive goals. NIH wants us to enroll over 2,000 participants by early 2026. So we have to think large and inclusive. They've mandated us to try to enroll as many or nearly all ALS patients in the country. We must increase the ethnic diversity in ALS clinical research studies. We must include people of various socioeconomic status and educational levels. So we have to really reach out. We have to reduce the barriers to participate, really engaging both the sites, but also individuals with lived experience with ALS, coordinate with all the ongoing studies that some of which I've listed already on a prior slide, and really enhance our outreach activities and communications across the spectrum in order to enable us to enroll this many patients this quickly. Next slide. And part of that is sort of decentralizing some of the methodology. So again, we're creating an all ALS website so people can actually go onto the website and identify themselves as being interested. Generating new tools of engagement in communities and outreach and recruitment materials. Using an e-consent for everyone within that participates. So you don't have to go into a site in order to enroll. A lot of either video conference visits, patient-reported outcomes collected remotely. We're doing speech initially, but likely other digital biomarker platforms will be included. And, as we're collecting blood, we'll have different cohorts, some that come onto our 34 sites, so we can collect site samples on site. Second, we're doing home phlebotomy nationwide. So sending registered nurses to homes to collect samples at the site of the participant. And if home phlebotomy can't reach the individual, well, we have a third option for a blood capillary collection that the individual participant can do on their own or with the aid of a loved one or caretaker when either the first two options are not available. So really trying to be inclusive so that everyone can participate. Next slide. So the initial two initial study protocols are in development and are currently at the IRB. And so one is called Assess All ALS and the other is Prevent All ALS. And next slide. So Assess is really the prospective observational study enrolling individuals symptomatic for ALS, so those with ALS, and also controls. Visits occur over a 2-year duration. And again, most of them will probably be remote. So they could be on-site or fully remote. Again, it's an observational longitudinal study collecting data, outcome measures, speech, and biofluids. And I've listed a few of these, again, clinical outcomes, patient-reported outcomes. Cognitive testing, we're using ECAS, which fits in well with our European colleagues. And then blood. CSF will be an option. Next slide. And the Prevent All ALS study is, again, another prospective observational study, but now we're enrolling individuals at risk for carrying inherited genetic variants known to be causative for ALS. And again, these could include C9 patients. And now the cognitive testing here, we've really reached out and we're working with our FTD colleagues. And so cognitive testing in this cohort could include ECAS, but also the cognitive testing that is performed for FTD patients. And again, collecting clinical outcomes, patient-reported outcomes, blood, and also possibly CSF. Next slide. So, conclusions. Tremendous progress on ALS biomarkers in this past decade. They're already making a significant impact on ALS drug development. Continued participation in ALS clinical research studies is really necessary to continue the development of these critical biomarkers. And now All ALS will hopefully enroll its first participants at the end of the summer and provides another opportunity, we think, for nearly everyone across the country to contribute to ALS clinical research. I'll stop here and happy to answer any questions.
Sorry, I was muted. We have a few questions for you already in the chat, so I'll read them to you. Are AI and machine learning techniques being explored to mine for novel biomarkers or composite biomarkers from the multiple biological measures?
Yeah, so the answer is absolutely. And, that's been done actually for many years. In ALS space, I could have been the first. I published a paper back in 2004 or 5 using machine learning algorithms and mining complex data sets. So AI approaches are a need, especially as we continue to develop more complex data sets that we want to analyze and correlate to clinical data.
Thanks. How do you connect the dots between understanding biomarkers and getting to therapy? Because in the end, that's everybody's goal, is to get to a therapy.
Yeah, that's a great question. And, it depends on sort of the approach that one wants to use. So for instance, in approaches that I've used, it's let's discover biomarkers. What's different between an ALS patient and control, a fast progressor or a slow progressor? And then you start to look at those biomarkers and start to see, here are pathways that seem to be changed. And then you ask, well, what are the drugs that are known to target those pathways? Have any of them been tried in ALS before? Have they been tried in other neurologic diseases before? Are they safe in humans? And so you can start to prioritize drugs that target pathways based on what's already known, but then take some of those drugs forward in drug development in ALS. So really identifying the pathways that seem to be altered based on the biomarkers, and then what drugs are available, or developing new drugs.
There's a question about whether you can have normal, I think, neurofilament light and still be symptomatic with ALS.
Yeah, it's rare, but it does happen. It usually, you can have lower levels of neurofilament in the blood. So the levels of neurofilament in the blood for an ALS patient are increased, but not as much as in the cerebral spinal fluid. And so you can have some ALS patients that when you look in the blood appear to have what should be for their age, a fairly normal level of neurofilament. That does happen. In the CSF, it's far more rare. Usually it's elevated in log order, so much higher levels than those in the quote unquote healthy population. So, but to address your question, often, when people ask me that, I'm thinking they're getting neurofilament assays of their blood. And so the differential between ALS and control is much smaller.
A question that came in earlier about whether the investigational biomarkers that are part of the HEALEY ALS platform trial, did they help inform the design for all ALS?
They did. I think the overall study design was based on HEALEY platform. Biomarkers being assessed in HEALEY platform will likely be assessed in the all ALS sample set. Again, some of this will come down to us from that AMP ALS steering committee. So we're poised with collecting all the samples, and that committee will help steer us in saying, all right, we should be testing neurofilament in every sample, or we should be testing, biomarker X in every sample. And then we'll have the funding to do that. So to answer your question, yes, both the HEALEY platform study and other prior clinical research studies in ALS were incredibly informative on the development of all ALS.
There's a question about prevent ALS, but maybe you can answer it for prevent and all ALS, of whether genetic testing is done to determine if someone's a gene carrier, asymptomatic.
Yes. Yeah, so the quick answer is yes, that will be done. I didn't have time to go into the real deep description of the protocols, but yes, there'll be genetic testing, there'll be genetic counseling services provided, et cetera. So all that is included in all ALS.
Next question might be more for Catherine, which is whether if somebody's on this webinar or goes to the HEALEY website or the NEALS website, can they find out about the studies that mentioned to participate?
Yes, so I work closely with Judy Carey at the HEALEY and AMG Center, who's the research access nurse for ALS trials overall. So if you're already in touch with me, I'm happy to help connect you. And I can put the webpage link in the chat here to where on our website, we post all ongoing observational and clinical trials for ALS that we know about through the HEALEY Center.
And another question for you, Bob, is whether low neurofilament predicts slow progression.
It does correlate to slower progression. Absolutely. So lower levels, again, they'll still be higher than normal, but lower levels do correlate with low progression. It's not 100%, but it is correlative.
I think we're at our time, 5:30. That was really wonderful. Thank you, Bob, for coming and sharing your knowledge and all these great studies. If people have other questions afterwards, please reach out either directly to Dr. Bowser or through Catherine Small.
Yeah. Thank you. Thanks, everyone.
Thanks. Bye. See you next week.